Hi guys, it's another exciting episode of Garage Laboratory. So this laboratory is a fermentation lab where we will set up kind of a makeshift fermentation chamber. Now to do this laboratory, we're going to be dealing with yeast. This is ordinary baker's yeast that we just buy at the grocery store. The yeast, we take a whole bunch of yeast and we put it into some warm water to break up the dried pellets into the individual cells. We have several clean test tubes that we're going to put the yeast into. We've got ourselves some water, and then we have various sugar sources. We have 5% glucose, 5% sucrose. We have the artificial sweetener sucralose. We have some cornstarch. We have some ordinary whole fat milk. And while it's not shown here, we are also going to utilize a poison for yeast metabolic activities. It's called sodium fluoride. The last thing you'll see here are a bunch of small plastic cuvettes that we've labeled up. We are going to fill these test tubes up with yeast and the various sugar solutions, and we're going to invert them in that plastic beaker. How you do it is a little bit of a trick. It takes a little bit of a coordination, but we're going to show you how it's done. As the yeast ferment, they're going to produce carbon dioxide, and we're going to measure how much carbon dioxide is produced. All right, so we're going to set up the last test tube. Dr. Martin's going to pipette out 5 mils of yeast solution. She's going to move it into the test tube. So what we have here is we have 5 milliliters of yeast in each one of these test tubes. Now we're going to add various various sugar solutions and water in order to fill up these test tubes. So Dr. Martin is simply going to fill up the first test tube, which is going to be our control, with water. You're going to fill it basically all the way up to the top. So this is what we're going to be adding. The next test tube we're going to fill up with a 5% glucose solution. The test tube next to that we're going to fill up with 5% sucrose. We're going to do 5% sucralose. We're going to do a 1% cornstarch, and then we're going to do milk. So here is our glucose being pipetted in. Again, you're just simply going to fill it up to the top. The next pipette test tube is going to be 5% sucrose. And then we'll repeat this using the cornstarch and the sucralose. And the last test tube is actually going to be a control in which we add the 5% glucose, and we're going to add some sodium fluoride, which is going to inhibit the activity of the yeast. So there are our test tubes. We've got ourselves the control, which is yeast and water. The next test tube, which is 5% glucose and yeast, sucrose and yeast, cornstarch and yeast, sucralose and yeast, milk and yeast. And then the final test tube you see here is glucose plus the poison, the toxin sodium fluoride, which will be a toxin or a poison to the activity of the yeast. Now we're going to set up the fermentation chamber. So we're going to set up a simple fermentation chamber in which we are going to take the test tube and flip it upside down inside each of these empty beakers. As fermentation proceeds, the yeast are going to break down the potential sugar source, producing carbon dioxide. Carbon dioxide is going to build up in the test tube, and it's going to push the liquid down into the beaker and form a carbon dioxide air bubble at the top. All you have to do is simply measure the height of the air bubble that forms over time. Now the tricky part is the flipping. But don't worry, since this is a virtual laboratory, I'm doing all the work for you. All right, so here's how we flip it. You simply put the beaker over the top of the test tube, and in one motion, flip it upside down, put it down. And the trick is make sure before you let go that your test tube is balanced. We're going to set the rest of these up, and as I said, as the yeast produces carbon dioxide, there's going to be a CO2 bubble that's going to form. It's going to push the liquid down into the surrounding test tube, and by measuring the height of that CO2 bubble, you get an indication of how much fermentation the yeast are performing. So there it is, guys. We've got all our fermentation chambers set up. This is going to actually run for a couple hours, and at certain times, we're just simply going to use a metric ruler to measure the height of the air bubbles. So that virtual data is going to be posted onto your Canvas page. All right, guys, we're back after a couple hours of these fermentation chambers running. So let's see what we have. We have our control and water. You can see there's hardly any CO2 formed. But if you take a look at the next two, glucose and sucrose, there is a significant air bubble, a significant CO2 bubble, which has been formed. Next to that, we have our cornstarch, which also has a little bit of an air bubble. Sucralose has some, milk has a little bit of an air bubble, and glucose with the poison sodium fluoride has virtually no air bubble whatsoever. Now what you're going to do to measure this again is you're simply going to take a metric ruler. Oh, here, Dr. Martin will take the ruler and she'll put it up against the air bubble and you are going to measure that. So it's kind of a guess, you know, like maybe that might be a 1/2 centimeter of an air bubble. And then you come down here and you put it right up against there, try and sneak it in. It does help actually to have a small ruler. Oh, perfect, there we go, measure that air bubble. And you would repeat for the rest of that. So again, this virtual data is going to be on your Canvas page.